My name is Micheal Gorka, and I'm a postdoctoral scholar at Penn State University. My research is on the biophysics of photosynthesis. It's an interesting topic because, on the macro scale, the conversion of light into chemical energy is responsible for virtually all life on Earth. But its success on the large scale is almost entirely due to how efficient the processes are on the small scale. Among the most interesting and efficient pieces of biological machinery involved in this process is photosystem 1. This photosynthetic reaction center is a protein that is embedded with a variety of what we call electron transfer cofactors. This includes 6 chlorophyll molecules, 2 quinones, and 3 iron sulfur clusters. Even though there are 2 active branches, let's simplify this down to one linear process. Now, the function of this protein is to convert light into a low potential electron that can be used for the fixation of carbon dioxide. When the electrons within the first chlorophyll are excited by light, the neighboring cofactor is just the right distance away to allow the electron to tunnel onto it. This tunneling process is then repeated until the electron has moved through to the other end of the protein, where it can be shuttled off to fix carbon dioxide. We know a lot about the processes that occur at the end of the protein, but we still don't fully understand what happens early on. We know that the initial stages involve these closely spaced chlorophyll molecules, but their exact roles, especially the one we call chlorophyll 2, has remained a mystery. Part of the reason is that these events happen on the order of femto and picoseconds, or a millionth of a billionth of a second, making them very difficult to study by traditional methods. Using a lot of light and low temperatures, we were able to isolate what we call the initial charge-separated state, and we knew the electron would be among these chlorophylls, and most likely on chlorophyll number 3. We analyzed these samples with a technique called two-dimensional hyperfine sublevel correlation spectroscopy, or 2D high-score. This allowed us to probe the chemical environment around the electron and essentially see what it was seeing. To our surprise, it wasn't just seeing one chlorophyll like we anticipated. Instead, we were seeing 2 distinct chlorophyll molecules. But the question remained, which 2? We were able to model this system using density functional theory, and we found that the electron was actually unevenly delocalized over both chlorophyll 2 and chlorophyll 3. This confirmed for us what had only really been speculated about in the past, that these 2 molecules function together as a single unit. This helped us solve one of the remaining mysteries of this protein, namely, what are the role of these 2 chlorophyll molecules? But more generally, it may have helped us change how we conceptualize the initial stages of electron transfer in these large photosynthetic reaction centers. We tend to think of this process as a discrete event, where an electron on some molecule A moves to B, leaving a positive charge on A and a negative charge on B, with the rate of this process being governed by things like the distance between the 2 molecules, free energy loss between them, and their reorganizational energy. But within these reaction centers, the early stages of electron transfer might better be thought of as a spreading of electron density through multiple cofactors, until the distance between the positive and negative ends becomes so large that we can consider the charges to be fully separated. And more than that, not all photosynthetic reaction centers share the same dimeric motif. And the question we have now is why? Why is this the case? What is the evolutionary advantage of these different chlorophyll motifs? Answering this question may help us better understand the history and diversification of natural photosynthetic systems, and we may even be able to apply these same design principles to our own artificial photosynthetic systems going forward.